Welcome to this Fast Facts presentation from Cell and Gene Therapy Insights and Refeyn Limited. I'm Abby Pinchbeck, editor at BioInsights, and today we'll be discussing how to quickly and accurately measure key analytical attributes of large vectors with the KaritroMP. Our presenter today is Jacintha Victoria, Market Development Manager of CGT at Refeyn Limited. Jacintha is a strategic marketing and analytical leader with over a decade of experience in the biotechnology and gene therapy sectors. In her role, she leads initiatives and campaigns to drive the adoption of mass photometry across critical applications in cell and gene therapy, including AAV, LVV, adenovirus, and RNA-based platforms. If you would like to ask Jacintha a question about this Fast Facts, simply use the Ask a Question box to the right of the viewing pane. And without any further ado, I'll hand over to Jacintha to begin the presentation today.
For today's presentation, I would like to introduce our latest mass photometry instrument called the KaritroMP. So the technology behind it and how we can utilize it to measure key attributes for larger vectors. So there are a number of critical quality attributes for viral vectors, but purity is one of the key attributes. This is mainly because presence of impurities in your product can significantly impact the quality of your product, efficacy of your product, and finally the safety of your product. Now, there are a number of analytical tools already available in the market for measurement of impurities, but they come with their own limitations. It includes the expense required to run the assay, the high expertise required to run it, the scalability issue, and finally, because it's a bulk measurement, the resolution with the individual populations. Now, there is a need in the market for a reliable, informative, fast, and an easy tool, and KaritroMP checks it all. So in scope of today's presentation, we would like to show evidence of how KaritroMP can be used to measure product impurities, can track or help with process monitoring, and finally can be an ideal tool for stability of the product. So to introduce our Refeyn family, we have two mass photometry units called the 2MP and SAMX-MP, both covering different size ranges as described below. Now, 2MP has demonstrated to be an ideal tool for characterization of proteins, antibodies, and nucleic acids, whereas SAMX-MP has been established for characterization of AAV serotypes. Now, with its latest GMP-compliant software release, now it can be adapted in both PD and GMP environments. Our latest addition to our portfolio is the KaritroMP, also called the macro mass photometry. We'll talk about the difference in just a minute, but this has been designed to target larger vectors such as adenovirus in the size range of 40 to 150 nanometers. So how does mass photometry differ from macro mass photometry? So mass photometry is a single molecule analysis tool which actually measures the mass of a particle with light. So as you can see in the image here, particles in solution are illuminated by a laser. Once illuminated, they scatter light and produce a contrast image, which is nothing but this darker spot you see on this image. So now, the higher the density or mass of the particle, darker the contrast, which means the contrast is proportional to the mass or density of the particle. Now, the software takes information, the contrast information for each of these particles, and segregates them into different populations, as you can see below. Now, the contrast information is converted into mass with a mass calibration. Now, macro mass photometry, in addition to the mass measurement done here, it does an additional measurement for size, which is called the Z-sweep or the vertical sweep. So what happens here is the Z-sweep helps define the diameter or size of the particle. So finally, the image you see on the right. So now you have a two-dimensional data segmentation, which is the size of the particle, and the contrast, which is the proxy for mass here. Now, with this information, you're able to distinguish the different populations going from small size, small contrast to populations in the same size, but different contrasts, thus giving a high resolution of population variations when you're measuring heterogeneous adenoviral populations. Here we looked at comparing KaritroMP to other commonly used methods in the industry for characterization of size distribution in samples. So for this experiment, we used NTA, which is nanoparticle tracking analysis, and DLS, which is the dynamic light scattering tool. We use the size difference, which is nothing but a mix of silica beads with 5 different diameters. So as you can see from the image, the DLS represented in green was only able to resolve one population. NTA represented in orange was able to resolve 2 populations. But neither of these 2 technologies, which follow the principle of Brownian motion, was able to actually detect all 5 populations in the mix, thus showing that these 2 methods actually have a bias towards higher size populations. Whereas Karitro, which is actually depicted in the different shades of blue, was reliably able to measure all of the 5 different populations in the size difference, thus concluding that Karitro, being a single molecule analysis tool, delivers high resolution without having the bias for larger size populations. So here we measured heterogeneous adenoviral samples on KaritroMP. So as you can see on this image, we measured a very low size and low contrast population, that is the fragment and protein aggregates. Then there were 2 other populations, which is the empties and fulls. So both have the same size, but they differ in contrast, mainly because of the density of the full capsids containing the payload. Now, quantification of these populations is key for process development and manufacturing of these adenoviral therapies. So Karitro can not only resolve these populations, but also quantify these populations reliably with just a single measurement, enabling reliable characterization of these diverse adenoviral populations. Now that we've demonstrated that the heterogeneous populations can be reliably measured in Karitro, we wanted to understand how accurate it is across the whole range. So for this experiment, we took empty capsids and full capsids and did different ratio mixtures going from 0% to 100% full and measured them on the KaritroMP alongside its orthogonal methods in the industry, which is AUC, SEM, ratio of UVs 260 and 280, and ratio of PCR over AEX HPLC. As you can see from the plot on the right, macromass photometry highlighted in orange has the best correlation fit between percentage foot theoretical calculated versus the percentage full measurement coming from the equipment itself. Apart from the reliability and the accuracy of the measurement, we also wanted to understand the logistics for using the technology to measure the impurities. So here in this table, we have the parameters going down, which includes cost per sample for running the equipment, the sample requirement, the speed at which it runs, and the throughput as the number of samples that can be run at the same time, the ease of use for setting up, running the experiment, and analyzing the data, and finally, the requirement for method development. And then going across on the top are the different technologies that we compared in the previous slide. The macromass photometry, or the KaritroMP, as you can see from this table, does stand out in all of the aspects mentioned below, thus helping us conclude that KaritroMP not only gives us reliable data as its industrial counterparts, but also helps us achieve that in a faster, easier, and a cheaper fashion. As we've already shown that it can reliably measure heterogeneous adenoviral populations, we wanted to understand if KaritroMP can be adapted as a process analytical tool for process monitoring. So in this experiment, we looked at different sample steps, as in like samples taken at different steps during the manufacturing process of an adenoviral drug. So we have samples taken from clarified, going from left to right. We have clarified sample, elution sample from a chromatography column, and finally your drug product. So as you can see here, your KaritroMP gives us an information on how the samples are depleted of the debris, which is the bottom left corner, which probably consists of your protein fragments and aggregates. So how that depletion happens across the different phases, and also the enrichment of your desired capsids, which is the full capsids at the drug product phase. So again, this shows us that KaritroMP, utilizing very minimal sample, and with its reliability of resolving different populations in a single measurement, can be adapted at a very early stage in the process for process monitoring and for process development. So now that we've established its reliability for heterogeneous population measurement, it being used or adapted as an early tool for process optimization, we wanted to look at other development workflows where KaritroMP can be utilized. So as we all know, one of the key aspects for vaccine and drug therapy is to understand or monitor the stability of your viral product. So here we adopted KaritroMP to be used as a stability monitoring tool for a sample that was heated at 47 degrees at different incubation time points. As you can see on the image below, on the right, going from left to right, you have the samples incubated for 0 minutes, 15 minutes, and 60 minutes. So it shows us that at 0 minutes, where you have an intact population, when the incubation time with temperature increases, you see a depletion or decrease in that intact population, whereas you have lower populations on the left bottom starting to appear, showing that or indicating that these are your broken capsids or broken intact particles. So again, KaritroMP has shown that it can monitor population changes, differentiating your intact population to broken down capsids, again, all in a single measurement within minutes. So yes, so how does KaritroMP's workflow look like? So we have the initial setup phase where it's sample prep, dilution, and loading. And this approximately is less than 20 minutes, and this is for up to 14 samples. So this is followed through with a 1.5-hour walkaway time, where all of the 14 measurements are automatically acquisitioned on the equipment itself. So because of this easy setup and workflow, it's easily adaptable and trained into every situation and every personnel. Finally, to summarize, so macromass photometry or KaritroMP is a single particle analysis tool, and here we've shown its reliability in being adopted to measure empty full fragment analysis, to help support process monitoring by being adapted in very early stage of the process, and finally for stability measurements for your viral vectors. And as highlighted in the previous slide, its intuitive workflow and its integrated automated data acquisition for up to 14 samples helps it to be easily adaptable into every situation and also be trained to new personnel. So overall, in a nutshell, KaritroMP is the ideal tool for characterization of large vectors and can all be done in a cheaper, easier, and a faster fashion. Thank you.
Thank you very much, Jacintha, for a great presentation today, and thank you to our audience for watching. If you'd like to download a poster version of today's talk summarising the main points, you can find that below the video. We hope you'll join us again soon for more Fast Facts.